Hello everyone. This video series is about bioinformatics, where we will be learning different topics related to bioinformatics like protein structure modeling, homology modeling, Ramachandran plot, how to interpret the data related to Ramachandran plot, then introduction to computer-aided drug design, computer-aided drug designing, computer-aided drug designing methods, then molecular mechanics and molecular dynamics. All these important topics related to this bioinformatics, and later we'll be having a quick introduction about docking and some important tools for molecular visualizations. So in this course we will be dealing with some practicals, but I won't be showing the practicals. I will be discussing about these practicals like homology modeling using SPDBV, Ramachandran plot using Swiss PDB viewer, then docking using August Lab, then active site determination by CAST-P, then small molecule building using ChemSketch, like that, then molecular visualization using RASMOL, then structural alignment using C-alpha-match. So these are very important and some sort of advanced topics. So today we'll be learning some important theoretical aspects related to the computer-aided drug design and introduction to computer-aided drug design. So computer-aided drug design includes pharmacogenomics and drug molecular design. So drug delivery is basically computer-aided molecular modeling. The molecular designing of drugs is based on knowledge of molecular properties like molecular structure, functional groups present, molecular geometry, etc. So as a process, drug designing is an interactive one involving drug discovery, lead optimization and chemical synthesis with the aim of maximizing functional activity and minimizing adverse effects. Then drug design does not include pharmacokinetics, dosage analysis or drug administration analysis. Only 1 out of 10 raised to 100 molecules possibly lead to a drug molecule. So hence a drug design is an optimization problem. So remember, only 1 out of 10 raised to 100 molecules possibly lead to a drug molecule. It is important to understand functioning of drugs in a general manner before getting into the molecular aspects. Like drugs can work in one of the following three ways, like interfere with the biological function of viruses, bacteria or parasites. All antibiotics fall in this category. Then another example is HIV drugs that try to interrupt the function of either the reverse transcriptase or protease enzymes in the virus. Then block interaction of viruses, bacteria or parasites within our system, and interfere with or enhance our own biological functions. Then enzymes catalyze biochemical reactions and they are a type of receptor like proteins. For example, digestive enzymes act to break down the nutrients of our diet. DNA polymerase and related enzymes are crucial for cell division and replication. Enzymes are proteins and are genetically programmed to be specific for their appropriate molecular targets. Enzymes ensure the specificity of their targets by forming a molecular environment that excludes interactions with inappropriate molecules. So lock-and-key hypothesis is used to explain the specificity of the enzyme mechanism. The enzyme is a molecular lock which contains a keyhole that exhibits a very specific and consistent size and shape. This molecular keyhole is termed the active site of the enzyme and allows interaction with only the appropriate molecular targets. The receptor is usually much larger than the active site. The receptor is specified by our DNA is folded protein whose major purpose is to form and maintain the size and shape of the active site. The most important aspect of drug design is the understanding of methods by which the active site of a receptor selectively restricts the binding of inappropriate structures. Any potential molecule that can bind to a receptor is called a ligand. For a ligand to bind with a receptor, it must possess the molecular key that binds the receptor lock. The molecular key consists of a specific combination of atoms that presents the correct size, shape and charge composition in order to bind and interact with the receptor. So ligand binding is based on the complementary relationship between the ligand and the binding site. This is a steric complementarity. The shape of the ligand is monitored in the shape of the binding site and physicochemical complementarity that allows molecular interactions between the two. So electrostatic complementarity restricts the binding of inappropriate molecules since the ligand must contain correctly placed complementary charged atoms for interaction to occur. Hydrophobic interaction drives ligand and receptor to interact, where there needs to be driving force that compels the ligand to leave the water and bind to the receptor. The hydrophobicity of a ligand is the cause for this. So when we talk about this, we need to learn different stages in the drug discovery process. So initial stage is research team formed and objectives are set. That will be the first thing involved in the preclinical studies. Then novel chemicals synthesized, later chemicals tested for efficacy and safety in test tubes and animals, then results used to choose drug candidates. Okay, later we will be performing chemical tests for efficacy and safety in test tubes and animals, results used to choose drug candidate. Then formulation, stability, scale up synthesis, chronic safety in animals. Then company files investigational new drug application with FDA. Then phase 1 studies in healthy humans. Then phase 2 studies in patients. Then phase 3 large clinical trials in many patients. Later, company files new drug application, that means NDA. Then FDA reviews NDA, then drug is approved for marketing. So these are some important steps used in the drug discovery process. Initially classified into two, like preclinical studies and clinical studies. Okay. Then we need to collect data. Then collaborator data is also needed. Then literature data we need, chemistry data, along with that biology data. All these data are, analyzed with the help of a computational chemistry software. Computational biology software is also needed. Then suggest better targets for intervention. They optimize ADMET bioactivity data, which we will be learning later. Then this prioritize molecules which are used in vivo techniques. So this is the virtual drug delivery process. So we need collaborator data. Later we need literature data. Along with that, chemistry data is also needed. Then biology data, that's obvious. All these data are combined with the help of computational chemistry software and computational biology software, which will definitely going to lead some results which suggest better targets for intervention. Okay, which are helpful for the conduction of in vivo tests. Okay, so computer-aided drug design, often called a structure-based drug design, involves using the biochemical information of ligand-receptor interaction in order to postulate ligand refinements. For example, if we know the binding site, the steric complementarity of the ligand could be improved to increase the affinity for its receptor. Indeed, using crystal structure of the complex, we can target regions of the ligand that fit poorly within the active site and postulate chemical modifications that lower the energetic potential by making more negative van der Waals terms, thus improving complementarity with the receptor. In a similar fashion, functional groups on the ligand can be changed in order to augment electrostatic complementarity with the receptor. So when a target is selected for the design of new lead compounds, three different situations can be faced regarding the amount of information of the system that is available. First, the structure of the receptor is well known and the bioactive conformation of the ligand is not known. And second scenario, only the bioactive conformation of the ligand is known. Then third one, the target structure and the bioactive conformation of the ligand are unknown. So the best possible starting point is an X-ray crystal structure of the target site. If the molecular model of the binding site is precise enough, one can apply docking algorithms that stimulate the binding of drugs to the receptor respective receptor site like O2 dock. So in the first step, the program creates a negative image of the target site through the use of several atom probes that determine affinity potentials for each atom type in the substrate molecule at different points in a grid. Place the putative ligands into the site, and finally they evaluate the quality of the fit. The program will try a set of different conformers of the ligand in order to obtain the best dispositions of the atoms of the molecule for maximizing the scoring function that quantifies ligand receptor interaction. A different strategy for obtaining new lead compounds through rational drug design is the de novo design of ligands with the use of a builder program like LigBuilder. This program also determines the shape and the electrostatic properties of the binding site cavity through the use of several atom probes, and then it combines from a library of chemical fragments those that better fit the cavity based on steric and electrostatic complementarity. Okay, so drug design approaches. There are different drug design approaches, like 3 basic approaches for designing drugs we'll be learning, like ligand based approaches, structure based approaches, and de novo approaches. Ligand based approaches in detail. These methods are used when the receptor is not known, ligand unknown, 3D structure generation, combinatorial chemistry. Then ligand, if that one is supposed to be known, like ligand is known. Then pharmacophore approach, QSAR we can conduct. Then database searching, etc. can be performed. So first let's learn about database generation. Combinatorial chemistry can be used to generate large libraries of molecules that can be screened for biologically active compounds. The use of combinatorial chemistry synthetic method allows a very large number of molecules to be synthesized much more rapidly at a lower cost than traditional synthetic chemistry. However, the enormous number and range of compounds that are produced using combinatorial chemistry are not necessarily accessible drug-like molecules. So ligand-based approaches try to identify characteristics common to known ligand to use in screening for new or improved drugs. If a set of active ligand molecules is known for the macromolecular target, but little or no structural information exists for the target, ligand-based computational methods can be employed. There are 2 methods to do this. Ligand-based computational. Ligand-based means either ligand unknown or not. So there are 2 methods to do this. Quantitative structure activity relationship, QSAR method, quantitative structure activity relationship. Then pharmacophore method. Next one is pharmacophore method. So quantitative structure activity relationship or QSAR. A QSAR study tries to establish a link between the ability of a certain molecule to perform its desired function and properties of that molecule. So QSAR method involves the statistical analysis of a set of properties or descriptors for a series of biologically active molecules. The statistical model developed is then used to predict the activity of additional compounds against the target. For example, the QSAR model can be used to predict which members of a series of proposed compounds are likely to be active and therefore should be synthesized and tested. As new compounds are assayed, the additional experimental data are used to refine the model. This approach has been used extensively in the pharmaceutical industry. Typical QSAR descriptors include physicochemical properties such as molecular weight, thermodynamic properties, and electronic properties. So clock P measure is also very helpful QSAR descriptor. So it is a good indicator of the compound's hydrophilicity. Low hydrophilicity is cause for absorption or permeation. It has been shown for compounds to have a reasonable probability of being well absorbed, so that their log P value must not be greater than 5.0. So besides these descriptions, you can also use statistical approaches for determining the model. This can include methods like least square optimization, principal component analysis, etc. Then pharmacophore hypothesis model. There are numerous potential interactions between ligand and receptor. Depending upon the size of the active site. There may be numeric, steric, electrostatic, and hydrophobic contacts. So, however, some are more important than others. The specific interactions that are crucial for ligand recognition and binding by the receptor are termed the pharmacophore. Usually, these are the interactions that directly factor into the structural integrity of a receptor or are involved in the mechanism of its action. If you consider the analogy of a lock and key, you need to imagine the lock having numerous tumblers. So there may be many keys that can complement the lock and fit within the keyhole. However, all but correct key will displace the wrong tumblers, leading to a suboptimal interaction with the lock. Only the correct key, which represents the pharmacophore to the receptor, contacts the appropriate tumblers and properly interacts with the lock to open it. It is important as a successful drug need to incorporate appropriate chemical structures and present the pharmacophore to the receptor. A ligand-based design by pharmacophore pattern is usually used when no receptor structure is available. Then target-based drug design. Target-based design methods bank on the structure of the biological target, either bound or unbound, to an inhibitor or substrate. So target-based design methods are also called as structure-based or rational design methods. If an experimental structure of a target is not available, it may be possible to create a homology model of the target based on the experimental structure of a related protein, which we will be learning later, homology modeling. So using this structure, candidate drugs can be predicted to bind to the target or designed. So alternatively, various automated computational procedures may be used to suggest new drug candidates. So if we summarize this, target-based methods are of two types, like ligand unknown and ligand known. Ligand known, for example, structure-based drug design docking. But if the ligand is unknown, we will be performing de novo design or a receptor-based 3D search. So docking method is the most popular approach for target-based methods. Docking involves scanning a database of known molecules for those likely to bind well to the receptor. Docking is used to generate possible binding geometries, and you can then evaluate using a scoring function. In docking, there are 3 degrees of translation and 3 degrees of rational freedom between the receptor and the ligand. 3 degrees of translational and 3 degrees of rational freedom between the receptor and the ligand. As this is already a very large space to simple, most docking methods to date use a rigid body assumption in the docking process. This is not a practical assumption in most cases, but it is made so that the problem will be somewhat tractable. Okay, so a docking method requires a representation of the receptor and ligand, a method for generating potential binding geometries, then a way of scoring these geometries. This method may also involve some refinement of the initially generated conformations before or after scoring. So today we just had a quick introduction about the computer-aided drug designing, where we learned important techniques, some introductory portion related to drugs and about preclinical trials, and then clinical studies, how to deliver a drug like that, and how to search the database. Drug design approaches, 3 drug design approaches like ligand based approaches, structure based approaches, de novo approaches, database generation, how to do that, then target based drug design, all these introductory portions. So if you have any doubts, please revisit this audio. Thank you for listening.